Hi all, I am Shisha Bose, a PhD student in Loughborough University. I am here to present on the topic Fracture Behavior of Collagen, Effect of Environment. Here is the outline of the presentation. Firstly, there is an introduction which is followed by materials and methods. Next, there is results and discussion, and finally we have the conclusion. Introduction. What is collagen? It is one of the most essential proteins which comprises one-third of human body proteins. It provides mechanical strength and structural stability to all living beings. It possesses a very complex hierarchical structure, with collagen molecules being lowest in the hierarchy. The collagen molecules bind up together to form the next level of hierarchy, which are the collagen fibrils. The collagen fibrils in turn bind up to form the next level of hierarchy, the collagen fibers. In this study, the authors are interested to look into the fracture property of collagen films. Collagen films find a wide range of applications, some applications being in tissue engineering, wound healing, flexible electronics, etc. Motivation of the study. It was observed during uniaxial tensile test that collagen films tested in aqueous conditions showed a drastic difference in mechanical properties as to those compared in air conditions. The figure shows the radical change in the stress-strain curves of in-air and in-aqueous conditions. In-air denotes samples that were tested in dry conditions, while in-aqueous indicates samples that were tested in biobath system. Now, in-aqueous testings are essential as they may partially mimic the in vivo conditions for potential biomedical applications. In the upcoming slides, all the in-air specimens would be denoted in red and the in-aqueous specimens would be denoted in blue. Hence, it would be interesting to investigate the fracture behavior of collagen films tested in air versus those tested in aqua. Materials and methods. Sample preparation. Collagen procured from bovine Achilles tendons were used to prepare solutions which were stored at 2 to 6 degrees Celsius for 48 hours. Next, the solution was stirred for a few hours, which was followed by centrifugation and degassing. Finally, the solution was casted and left in fume hood to dry. After a couple of days, a nice, easily peelable collagen film was obtained. For fracture testing, single-edged notch tension specimens, sand specimens were prepared. The gauge length and width were kept at 20 mm and 22 mm respectively. A sharp notch of 11 mm was introduced with a scalpel on one edge of the specimen, keeping a ligament length of 11 mm. Materials and methods. Experimental setup. The whole experimental setup with the biobath system is exhibited in this figure. A camera was placed perpendicular to the samples so as to record the changes in the deformation behavior during the entire process. A zoomed-in view of the grips is also shown. In the upcoming slides, collagen films, those were tested in air, would be designated as COLD, while those that were tested in aqua would be designated as COLA. In aqua samples were prehydrated for 10 minutes before testing. Results and discussions. Stress strain curves. COLD films behaved more like a non-ductile material with less capability to deform plastically. This resulted in a low strain at failure of about 3%, as presented in the table. COLA films under the influence of water exhibited a non-linear stress-strain response with an increase in strain at failure to about 51%. In aqua conditions is responsible for extensive plastic deformation in collagen. The ultimate tensile strength dropped to 0.96 megapascals in COLA films as compared to 19.5 megapascals in COLD films. The fracture energy also reduced by 40% for in aqua conditions. Thus, it might be said that in aqua, the ductile deformation is enhanced. However, it is also accompanied by a severe reduction in strength and fracture energy. Results and discussions. The analysis of crack profile or deformation behavior for in-air and in-aqua conditions would be of high interest. Thus, the deformation behavior of COLD is presented in this slide. The schematic represents the direction of application of force. This figure depicts the initial pre-crack introduced in the specimen. With the increase in tensile deformation, the crack propagation begins. The crack propagates rapidly with very little plastic deformation, which is accompanied by a brittle failure. Hence, it might be said that in-air conditions lead to a rapid brittle crack propagation in collagen films. Results and discussions. Deformation behavior, COLA. The schematic represents the direction of applied force in COLA specimens which are subjected to in-aqua loading environment. This figure represents the initial pre-crack introduced in the specimens. As the tensile deformation begins, initially crack opening takes place at a very slow rate. This is then accompanied by crack blunting with a rapid crack propagation until failure. Hence, in aqua conditions, there is a gradual crack opening and propagation with an intensive plastic deformation as compared to in-air specimens. Results and discussions. Estimation of fracture toughness. The sudden rapid crack propagation of COLD films led to brittle failure. Thus, for COLD, the fracture toughness parameter K1C was evaluated using linear elastic fracture mechanics (LEFM). K1C can be estimated using the following equation, where P is the force applied, W the width of the specimen, B the thickness of the specimen, A the pre-crack length, and function of A by W the dimensional constant, which depends on the shape of the specimen. K1C denotes the critical stress intensity factor of mode 1 loading. The K1C for COLD is around 17.5 megapascal into millimeter to the power half. In COLA, there was extensive deformation accompanied with slow crack opening and propagation, leading to a ductile failure. Hence, elastic plastic fracture mechanics (EPFM) was considered to calculate the fracture toughness parameter JC integral, which denotes the critical J integral. JC may be estimated using the following equation, where P is the force applied, delta the displacement, B. The thickness of the specimen. Small b. The ligament length. Eta. The dimensional constant which depends on the shape of the specimen. Jc for COLA is about 2.5 megapascals into millimeter. Conclusions. Three basic conclusions can be drawn from this study. A drastic drop, approximately 40%, was observed in the fracture energy when collagen films were tested in aqua. Hence, while designing collagen-based materials, all the mechanical testing should preferably be performed in vivo conditions. Second, analysis of deformation behavior in air and in aqua conditions confirmed that linear elastic fracture mechanics (LEFM) may be applied for collagen tested in air due to its brittle failure mechanism, while due to slow crack opening and blunting mechanism of collagen tested in aqua conditions, elastic-plastic fracture mechanics can be used to estimate the fracture toughness parameters. Third, the fracture toughness parameters is reported here. K1C about 17.5 megapascals into millimeter to the power half for in-air specimens, while JC is about 2.5 megapascals into millimeter for in-aqua specimens. Thank you.